Bacteria have to regulate their metabolisms in a lot of different ways in order to properly adapt to their environments. Because they don't have eyes, ears, or any of the senses we do, they have to come up with lots of other ways to determine things like how much light there is and how many other bacteria are around them. In this video, we'll discuss one of the mechanisms by which they do this, quorum sensing. We'll cover what it is, how it works, and why it is so important. The name quorum sensing comes from the concept of a quorum, which is the minimum number of people required to be present for a meeting before business can be conducted or decisions can be made. Similarly, quorum sensing is a way through which bacteria can regulate their behaviors or metabolism to only do something if enough bacteria are present. There are lots of situations in which this could come in handy. For example, an invading bacterium in your bloodstream might want to go unnoticed by your immune system until it's present in large enough numbers to have a chance of surviving, so it uses quorum sensing to only produce toxins once there are enough bacteria there. Quorum sensing could also come in handy for the formation of a biofilm, which is essentially a community of different species of bacteria growing in a sticky matrix cooperating to survive. By using quorum sensing to recognize when enough other bacteria are present, each individual bacterium can save a lot of energy by only deciding to turn on the genes for producing that sticky matrix or making certain chemical compounds when there are enough of them around to make it worthwhile. So how does this all work? How exactly do bacteria accomplish this unified change in gene expression to produce such dramatic results? To answer these questions, let's look to one of the most well-known and beautiful examples of quorum sensing, the partnership between the bacterium Vibrio fischeri and the bobtail squid.
As mentioned by Alex Padron in their article Quorum Sensing Microbes and Squids published in 2013 in the Berkeley Science Review, the Hawaiian bobtail squid uses bioluminescence to protect itself from predators. In other words, it glows in the dark. The squid has several light organs inside it that house the bacteria responsible for making that blue glow, and the light produced has the dual function of eliminating its shadow and also making it difficult to see against the bright moonlight coming down from above. In the day, when the squid buries itself in the seabed to sleep, it squeezes out about 95% of the bacteria from its light organs and the glow stops. By the time night has fallen again, the bacteria have grown once more to a high enough density that the squid again begins to glow. It's worth noting that V. fischeri only glows when it's inside the squid. Even though it lives free-floating in the ocean, the only time it's at a high enough population density for quorum sensing to actually trigger light production is inside the squid's light organs at night. So how do they tell? Let's look at the molecular background. Quorum sensing works through the production of chemicals called autoinducers. These chemicals work just like any other compound that induces a gene to turn on, but they're termed autoinducers because they stimulate genes in the same cell that makes them. In the case of Vibrio fischeri, the gene that makes the autoinducer is called LuxI, and the receptor protein that detects the autoinducer is called LuxR. The cell constantly produces LuxI, which diffuses out of the cell and into its surroundings basically immediately after it's made. However, if there are enough cells nearby also making LuxI, the concentration of LuxI outside the cell accumulates to such a large concentration that it starts diffusing back in, attaching to the LuxR receptor and forming a duplex molecule that stimulates the expression of the genes that make light.
The thing is, many bacteria use quorum sensing as a regulatory mechanism, and so there are a lot of different autoinducers floating around in solution. As it turns out, some bacteria make the same autoinducer as other types of bacteria, which essentially allows communication across species through the quorum sensing mechanism. Yet more bacteria produce receptors for competitor organisms' autoinducers, allowing them to sense the population density of other species nearby. Some species even produce chemicals that bind another species' autoinducers and prevent them from diffusing back into the original cell, or bind their receptors directly and downregulate instead of upregulate gene activity, essentially using quorum sensing mechanisms to sabotage the growth of their competitors. It's like an entire language of bacterial regulation, using chemicals in the environment to send messages that can dictate the behavior of thousands of other species. It's easy to see how, by learning to speak this bacterial language, we can tap into their communications and modify their behavior. In the case of bacteria that rely on quorum sensing to tell them when to produce toxins that make us sick, we can manufacture an anti-autoinducer that would block their abilities to read messages from other bacteria, meaning that they would never receive the signals telling them to start producing nasty byproducts, thereby giving our immune system time to fight them off. Or in the case of biofilms, blocking quorum sensing could help remove or even prevent entirely the formation of that nasty scum on surfaces submerged in liquid. For example, the surface of your teeth or the tiles of a swimming pool, as discovered by Subhadra and Associates in their 2018 work, Control of Biofilm Formation in Healthcare. Understanding quorum sensing so that we can best exploit it is thus becoming increasingly important in the modern scientific and medical world, especially as we have to come up with increasingly creative ways to solve the problems we encounter on a larger scale. Maybe as our comprehension of this phenomenon becomes more nuanced, we'll be able to harness bacteria to do things we haven't even dreamed of yet.